Hello everyone and welcome to DDL 2021. My name is Raquel Borda Água and today I will talk about the work we've been performed regarding leveraging DPI formulation screening. Just before I start with the technical part about my presentation, let me take a moment and introduce the company where I work. Ovion is a contract development and manufacturing company dedicated to helping pharmaceutical customers bring new and off-patent drugs to market. Ovion has over 60 years of experience in the development and compliance manufacturing of APIs and drug product intermediates. The topic that I'm going to talk today is dry powder inhalers, and the dry powder inhalers are the devices most commonly used for the drug delivery in the treatment of asthma and pulmonary disease, and have attracted huge attention all over the world due to its local targeting, rapid drug effect, and reduced systemic toxicity. However, dry powder inhalers formulations consist of highly cohesive powders that tend to agglomerate, and for that reason, fine and coarse carriers are often used to reduce the cohesion and to promote the flowability and the aerosolization. So, understanding the role of the cohesive adhesive forces in different formulations and establishing a predictive approach for aerodynamic particle size distribution is highly beneficial since the traditional next generation impactor is a complex and time-consuming technique. The purpose of this study was to explore the relationship between powder dispersibility with the aerodynamic performance of different dry powder inhaler formulations, and for that, three different carrier-based DPI formulations were selected using coarse and fine lactoses with different ratios. And regarding the characterization, the morphological analysis of formulations was carried out using scanning electron microscopy. The aerodynamic particle size distribution was measured by NGI, and the aerodynamic properties such emitted dose, fine particle fraction, fine particle dose were calculated, and the inherent powder dispersibility and the agglomeration was assessed using a laser diffraction technique. In this case, we used Sympathec. As I mentioned before, microscopy was used to investigate DPI's morphology and state of agglomeration, and this figure shows that coarse lactose was covered by API and fine lactose. And if we think on particle shape, coarse lactose shows a rock-like shape morphology, while API and fine lactose particles showed an irregular form, being localized on the surface of the coarse lactose for all formulations. Nevertheless, a different distribution of particles on the top of the coarse carrier was observed between formulations A and the other two. A higher amount of particles can be observed on the top of coarse lactose for formulations B and C. And as you can see, the increase of API and fine lactose lead to more agglomerates and API fine lactose on the coarse lactose surface, presenting larger agglomerates on the top of the carrier compared to when fine lactose was almost absent in the formulation. On the left side of this slide, we have represented the aerosolization response for the three formulations that we use in this study. And in this table it can be seen that for higher drug content and higher fine lactose formulations, fine particle fraction values increased, implying that the increase of the drug content and the amount of fine lactose could be an effective way to enhance lung delivery by helping drug detachment from the carrier surfaces. But since the goal of this study was to understand the deagglomeration and the aerosolization mechanisms, the same formulations were investigated using laser diffraction technique, and the X10, X50, and X90 parameters were plotted as a function of the pressure, but only X10 and X50 parameters are represented in the figures on the right side of the slide. And as can be seen, all the powders exhibit a reduction in these parameters until a plateau was reached, and the shape of the particle size pressure profile showed a rapid drop in X10 for formulations B and C under the application of a dispersing pressure of 0.5 bar, which indicates an efficient agglomeration when compared, for example, with formulation A, where the drop in the X10 and X50 was more progressive for this formulation, suggesting a more gradual deagglomeration process. And the same tendency was also observed in the X50. The decrease observed from 2.5 to 4 bar could due to the potential milling of particles. No significant differences were observed in X90 between formulations, since this parameter is highly influenced by coarse lactose and for that reason was not used for aerodynamic performance evaluation. This result seems to show that formulations with faster deagglomeration behavior, the agglomeration that occurs at low pressures, also have higher fine particle fracture values, and this finding supports the theory that the agglomeration assessed by laser diffraction using Sympatec could be correlated with the aerodynamic performance. By comparing the aerodynamic particle size distribution results obtained through NGI and the ones obtained by laser diffraction methodology, it is observed that the formulation with the higher amount of API and also the higher amount of fine lactose has faster deagglomeration. It is also the formulation with the highest fine particle fraction value with 27.6, which allows to conclude that the method may have the potential to estimate the aerodynamic performance during DPI screening process in the early development stages. In a matter of conclusion, after this study, a proof of concept is presented, demonstrating a trend between laser diffraction results with aerodynamic performance for a micronized API in three different carrier-based formulations. The results showed a rapid and systematic laser diffraction methodology enables the prediction of aerodynamic behavior, accelerating the screening process of inhalation formulation, and currently further studies are ongoing with different formulations and APIs in order to statistically infer the forecasting ability of Sympatec in supporting pre-formulation development. Thank you for your time. I hope you all enjoy the session.